Good day, says Professor Will, CBMD PhD. Today is Monday, October 18, 2021. It is 9:40 p.m. Eastern Standard Time. Today's research, photoreceptors. So let's hop to it here. Developing a treatment for vision loss through transplant of photoreceptor precursors. Date October 18, 2021. Source, National University of Singapore, Yong Lu Lin School of Medicine. A recent study examining the therapeutic potential of photoreceptor precursors derived from clinically compliant induced pluripotent stem cells, iPSC, has demonstrated the safety and therapeutic potential of clinically compliant iPSC derived photoreceptor precursors as a cell replacement source for future client trials. While the introduction of gene therapy has been a significant development, its effectiveness has been blunted by the sheer extent of genetic heterogeneity with more than 260 genes implicated in IRDs, and this limits the widespread application of gene therapy for all IRDs. Additionally, gene therapy has limited efficacy in clinical cases of advanced retinal degeneration, which significant photoreceptor cell death has already occurred. And photoreceptor cells are found in the retina and respond to light, converting it into electrical signals that activate physiological chain reactions. These signals are sent through the optic nerve to the brain for processing. And with the advent of induced pluripotent stem cell, iPSC, and embryonic stem cell ESC technology, regenerative stem cell therapy has the potential to be an alternative treatment for end-stage retinal degeneration, independent of the underlying genetic defect. Retinal regenerative therapies therefore hold great promise for the treatment of IRDs. Studies in animal models of IRDs have suggested visual improvement following retinal photoreceptor precursors transplantation, though there is limited evidence on the ability of these transplants to rescue retinal damage in higher mammals. Researchers examined the therapeutic potential of photoreceptor precursors derived from clinically compliant iPSCs. The study demonstrated the safety and therapeutic potential of clinically compliant iPSC-derived photoreceptor precursor as a cell replacement source for future clinical trials. Good day, says Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.